The overall goal of this procedure is to demonstrate the technique of RNA interference in ticks by injection. This is accomplished by first synthesizing double-stranded RNA of the gene or genes of interest. The RNA is then injected into ticks, after which they are held in a humidity chamber for 24 hours. After this holding period, the ticks are allowed to feed on an animal, such as a sheep. Finally, tick feeding parameters, such as weight, molting, and oviposition, and the expression of the targeted gene or genes of interest by real-time RT-PCR are determined. Ultimately, results can be obtained that demonstrate the effect of silencing targeted genes on tick biology by evaluation of tick biological parameters and gene expression by real-time RT-PCR.
Our group works with ticks, trying to characterize molecular events at the tick-pathogen interface, to use this basic information to develop methods for control of tick infestation and the transmission of pathogens to humans and animals. RNA interference has emerged as an essential tool in this research because it is the only method available for genetic manipulation of ticks.
The function of many tick genes is unknown. RNA interference allows us to define the role of tick genes and gene expression in many aspects of tick biology, including mating, reproduction, feeding, digestion, salivary gland function, and the tick vector competency for the transmission of pathogens. Demonstrating the procedure will be the tick RNA interference team from our laboratory, Dr. Ed Blouin, graduate student Ann Busby, and myself.
To generate double-stranded RNA, first synthesize oligonucleotide primers containing T7 promoter sequences for in vitro transcription of RNA. For example, for